heard an inspiring talk from Trevor, but we get to ask him a few more questions in this, but this is a change in format. So we have 5 distinguished panelists, and I'm going to ask each of them in turn to say a few words about who they are, how they got to their current position, and what their view is of the way they represent their institution's goals, and in a sense why they're here at this symposium today. And I hope we'll leave time for questions and interactions with the audience. Just to remind you, I'm Chris Walsh. I'm on the board of directors of Scripps in my lifetime. I've done, I think, 30 biotechnology companies since 1981. These days, I'm at Stanford, where I'm part of the Chemistry, Engineering, Medicine for Human Health initiative. I'm also a senior advisor to the Chan Zuckerberg BioHub initiative in San Francisco, and I'm a great believer in what the Scripps caliber people are trying to do. Sir.
All right, okay. So I'm Brian Fiske. I'm a Senior Vice President of Research Programs at the Michael J. Fox Foundation for Parkinson's Research. I've been there since about 2004, which is just shy of a couple of years. They were founded in late 2000 by the actor Michael J. Fox, and then started hiring some in-house scientific staff around 2004, so that's when I came on. And I've been there ever since, really helping them sort of shape and develop their research efforts and research programs. So our mission is obviously by our name and our founder, obviously pretty clear. We're focused on Parkinson's disease, and really through our activities focused on supporting and accelerating research and drug development that can lead to improved treatments and ultimately cures for Parkinson's. Don't know how much background everybody has on Parkinson's disease. Obviously, it's a very disabling and troubling disorder, age-related neurodegeneration, primarily due to loss of dopamine-producing neurons in the brain, but a whole host of other symptoms and sort of pathology that can lead to really troubling symptoms for people with the disease. Our focus is always really been, again, on identifying the key challenges in that sort of therapeutic translational pipeline. So from the early sort of disease biology space, what targets, what mechanisms we think ultimately are disrupted and altered in Parkinson's that we can then ultimately help and support drug development around to develop treatments that can sort of manipulate that biology and that pathology and ultimately lead to cures. So it's a pretty complex mix of what we support, again, from early discovery, early biology, preclinical development of therapeutics and testing of those therapeutics in preclinical models, all the way to early stage clinical trials. And that's something that we've been able to do in the last few years as our fundraising capacity has increased and our ability to operate in that space has become sort of more capable. Not just through funding mechanisms, we support a lot of different approaches and other ways of sort of addressing challenges in the pipeline. For example, we are very hugely invested in around biomarkers and sort of better outcome measures, whether it's from sort of more traditional biochemical markers all the way to sort of more patient-reported and digital types of biomarkers as well. Really with the goal of you could have a really great drug, but if you can't really measure the impact it's having on the disease biology itself, then you're not really going to learn much in your clinical trials. We've also been very big on sort of educating the patient community and the community at large in the need to get involved in research, so not just more for general advocacy, but the actual need we have for them to participate in the challenge. And so we've done a lot of work, for example, over the last few years in sort of addressing issues around recruitment challenges in Parkinson's disease clinical trials and making sure that we can find the right people to be in those trials and make that whole process sort of more accelerated and more informative. So maybe I'll stop there.
Thank you, Brian. Ramy? You may each have a microphone, or perhaps not. Okay, good.
All right, good afternoon. So I'm Ramy Ibrahim. I'm the Chief Medical Officer at the Parker Institute for Cancer Immunotherapy. I've been with the Institute almost from the very beginning. I always think of us as the new kid on the block because we are a relatively young organization. We just launched in April of 2016. By background, I'm a medical oncologist, and like many immunotherapists, I started my career contributing to the number of negative cancer vaccine studies that were done at the National Cancer Institute and other places. But this really got me more interested in trying to understand the immune system and how we can use the patient's own immune system to fight cancer. After the NCI, I moved to pharma, and then I attended the launch meeting for the Parker Institute. And when I heard Sean Parker talking about his vision and how he is really wanting to start something that's relatively disruptive, where he wants to bring different academic centers and provide the infrastructure that can allow the scientists from academia to be able to collaborate and advance their discoveries. So I think there was a question earlier this morning about the difference between pharma and nonprofit. For me, that was the biggest factor that made me very easily leave my career in pharmaceuticals and move to the nonprofit. Just that goal of being able to take a discovery and help a scientist advance it until it becomes something that can actually be used to treat patients. At the Parker Institute, again, our model is slightly different than some other nonprofit organizations because we are focused on a set number of cancer centers, and we started small because we wanted to make sure that the model works. We wanted to show that we can actually build this infrastructure that can allow people from different academic centers to freely collaborate together, share data, share findings, without compromising their ability to publish, without compromising their ability to file any intellectual properties that they want to file on their discoveries. So right now we have 7 academic centers in the US that are under the umbrella of the Parker Institute, and those are Dana-Farber, which was recently added to our network. But we started with Memorial, Penn, MD Anderson, UCSF, UCLA, and Stanford. But we're not limiting our support just to scientists from those 7 academic centers. We're constantly looking for other scientists at other institutes that might have disruptive ideas, and they are looking for the support, and we try to give them this support. So now we have members from Wash U, from Mount Sinai. We even started talking with other centers outside of the US, because obviously some of the feedback that we heard early on was that there are smart people outside of the 7 academic centers, but obviously we are limited to, or at least we wanted to start small, but we are talking with other groups outside of the US to make sure that if there are projects that we can collaborate around, that we can do it. Sean's vision, again, when he founded the Institute, was he wanted us to be very focused. So again, that's why our name is very long, but he wanted to make sure that it's cancer and it's immunotherapy. So those are the 2 things that we are focused on. So as long as it falls under the cancer and leveraging the immune system, then we'll be interested to talk with the scientists about their discoveries. The way that we provide support is not just giving out grants. So we never ask for RFPs and for people to come forward with proposals. What we're really doing is meeting with our scientists, making sure that they are talking to each other. And once they identify a project that they want to collaborate around, we do provide some funding to support this, but I think our contribution is more than just the funding. Our contribution is we have a clinical trial management group, so it allows us to take ideas and turn them into an actual clinical trial that can be done across multiple institutes compared to what's usually happens with academic centers, that they do single institute investigator-sponsored study that's usually small in size and has limited resources. So by having the clinical trial management capabilities, now we can hold INDs and run multi-center clinical trials, do it efficiently, but at the same time, do it with a quality that meets industry standards. Because the problem that we're trying to solve is that once we have a finding from some of those studies, we want to make sure that a pharmaceutical company can take this and run with it, because they know how to do registrational studies, which is something that we don't do. We have the translational capabilities. We're trying to bring newer technologies that we can make available to our members beyond what they already have in their own labs. And that requires us to partner with many startups and many technology companies that have maybe platforms that are still not fully validated. So we help them to do the validation, but at the same time, we give access to our members to those newer technologies that can allow them to better understand the data that they are generating, whether it's preclinical or clinical data. We have a bioinformatics group, and this group is really helping the scientists learn more from either existing data or new data that they are generating. So some of the support that we give to our members is just asking them to give us data that they've already used, published, patent, they did everything that they can do with that data, and we just tell them, give it to our guys and let them take a look at it. And they really start doing data integration and deeper data analysis. And actually, we did this the past couple of years, and it ended up with having discoveries that made it to Nature and Science. So we're not talking about like small discoveries, but it really shows the strength of doing a deep data analysis versus just here's a discovery, here's a paper, and here's an IP, and let me move to the next project. And then the last piece that we do is the business development part, where, again, many scientists, they always feel that the end goal is either a science paper and an IP, and then, as I said, you start doing the next experiment. What we're trying to help them with is really understand that science papers are great, but they don't cure cancer. So for us, having a science paper is just the beginning of actually the path to try to find a new cure or treatment for cancer. So we have a group that can help scientists take their discoveries and turn it into an actual company that can start developing this and moving it forward. We can give them some seed funds that can allow them to do some very focused preclinical work that would inform the IND. We can help them put their data package in a format that will the VCs and other investors can relate to versus just a scientific slide deck. So really put them in the first or in the right path to start having a company that can eventually take this discovery and continue the development.
Thank you, Ramy. Maria.
Hello, I'm Maria Millan, the president and CEO for the California Institute for Regenerative Medicine. So CIRM is similar to the previous two speakers. It is dedicated to funding basic and translational research. It's unique in that it is a state bond initiative that was used to create the agency, a $3 billion bond initiative that was passed by the voters of California to support stem cell regenerative medicine research just years after the isolation of the human embryonic stem cell, because those voters who supported it did not want science to stall as things were getting figured out from the policy side. And at that time, there were federal restrictions on the generation of human embryonic stem cells. And then, of course, subsequently, the identification of the induced pluripotent stem cells, and now CIRM had actually funded the creation of the largest iPSC bank that you can all use for your work, over 2,000 lines from a variety of disease indications and targets. So Broad is using quite a bit of them and other investigators around. For that $3 billion, we're now, it started 2004, it stalled for a couple of years, but since then, we funded 1,000 projects and now over 51 clinical trials. And just to speak to the maturation of the field, in just the past 3 years, we've tripled the number of clinical trials from that which was just going into the clinics in the first 10 years of the agency. So we now have funded over 51 clinical trials, and it's in a variety of disease indications, from rare diseases such as fatal diseases like alpha thalassemia, severe combined immunodeficiency, 3 forms of it, in fact, as well as more common diseases like diabetes, osteoarthritis. One of our programs that was featured earlier today is a CIRM partnership with Caliber and Scripps. Very excited about that, as well as really kind of unique approaches to various unmet medical needs. So the requirements, or what CIRM does is we de-risk high-risk, high-reward programs. Earlier, Dr. Rosen spoke about the collision of orbitals, and it's in the execution that we could really impact the probability of success. And so what CIRM has done in terms of the execution has created an acceleration model that allows us to sort through high quality science, identify the best science. We have recurrent and predictable funding, always open for clinical stage and preclinical stage programs, so people don't force fit their applications. We are partner along the way with our investigators to take a look at their value proposition. Our review panel, our what we call grants working group, composed of, is derived from a panel of over 200 highly regarded specialists, and each review includes 15 of those 7 patient advocates. So from that, what happens is we de-risk based on quality, and we fund early stage programs where industry and traditional investigators, traditional investors would not fund. What that's resulted in is that we've been able to, for the $2.6 billion that we've allocated so far, another $3.2 billion has come in either follow-on funding from investigators, philanthropists, and other sources. Just in this past year alone, more than $1.06 billion had come in from partnerships for these programs, including programs that came all the way from the bench, like CD47 out of Stanford, now into clinical trials in liquid and solid tumors, like Don Cohn's program from UCLA with a gene therapy for ADA, adenosine deaminase deficiency, SCID, now is in pivotal phase 2B trial. We also, in terms of being a translational machinery, are very well connected with our investigators as they navigate a very new pathway toward regulatory initial trial to approval. And you may know that the 21st Century Cures Act, which was just passed December 2016, created an expedited pathway in the FDA specifically for regenerative medicine. Of the first 3, 2 of them were CIRM programs, and of the first 25 of them were CIRM programs. So it's really a very unique position to be in, to be charting the way. And the cool thing about this pathway is that it recognizes for stem cell regenerative medicine that it has very unique qualities that are distinct from small molecule and traditional biologics in terms of mechanism of action. Generally is what drives the development. Hard endpoints are not PK/PD, they're often readouts, efficacy readouts. Now surrogate endpoints and biomarkers are becoming more prominent. Use of registry data becoming more prominent. In fact, at the same time that Kymriah was approved as the first gene therapy, CAR T therapy, the blinding eye disease therapy for a monogenic disorder, Spark Therapeutics, was approved based on, number 1, completely novel endpoints using a maze functional study, and 2, utilizing registries. So, and in terms of industry engagement, we actually have an industry alliance program which helps to really bring in industry input and potential partnerships early on in the course of development of these programs. I think that's all I want to say for now.
Trevor, you have a chance, in case you've rethought some of the things you've said, or a reprise, an opportunity to mention something you didn't get to say in your earlier talk.
Right, thanks, Chris. And I think that folks know my story, and Pete gave a good account of the somewhat tortuous path that I took to get here. So maybe just a few other comments. So Bill and Melinda, based on their tenet that all lives have equal value, together with Warren, now allocate about $6 billion a year to this enterprise of theirs, which is partly in global health, development, also in education. So, I've been privileged to be part of that enterprise and be able to deploy some of those resources into the global health space. And I would say, just in terms of the nature of that relationship and how that's evolved. So Bill has these sort of twin characteristics. On the one hand, he has an abiding fear that there is some invention out there and some technology that we don't know about. And he never sees a new technology that he doesn't like or is interested in. So that sets up one set of parameters in terms of the work we do. On the other hand, he's a very pragmatic businessman. So he wants to see results. And he wants to see things actually happen as opposed to just theorize and talk about. So we have this tension, and I like to think of it as a very productive tension, which many of you have experienced in terms of how do you advance cutting-edge innovation, which almost always fails in its first incarnation and needs to be iterated, at the same time as how do you actually have practical results. And that's the debate that I somewhat started at lunch, and am very interested to hear in other ideas from you folks. Thank you.
Thank you. Paul, you are sitting amongst 4 foundation and nonprofit groups, and you are bringing a Vertex perspective, but of course, you guys have interacted with the Cystic Fibrosis Foundation in novel and very productive ways.
Well, thank you for the invitation to join the panel. My name is Paul Negulescu. I'm Vice President of Research at Vertex. Vertex has only been profitable for 3 or 4 years of its 30-year existence, so I'm almost qualified to be on your panel. I think one of the reasons I'm here
It's time
today,
for you to be philanthropic.
It is for sure. One of the reasons I'm here is that Jim Schaefer called me up a few months ago and indeed asked me if I would be willing to share some perspectives on the public, on the partnerships between pharmaceutical companies and not-for-profits. And I see Chris Penland is here a few feet away from me, who I've worked with at the CF Foundation for almost 20 years. He was involved in the initial decision to grant then Aurora Biosciences an initial grant to work on CF. It's an example of a successful partnership, of course, because out of it came 3 approved drugs that are indicated for approximately 50% of the population. And we have additional compounds in phase 3 studies that could treat up to 90% of CF patients at targeting the root cause of the disease, targeting the underlying protein defect. So that was a project that in the 1990s, when we started, was given a very low chance of success because there had never been a small molecule that could modify a disrupted protein. I think now we know that small molecules can do incredible things, and we've seen some good examples of that today, but I think back in the 90s it was sort of unprecedented to try to correct proteins in this way. So I think we've all learned that the impossible can be possible, and that partnerships with patient groups can be very, very productive. I'd say that one of the things that really, and we can talk about it more later, but one of the things that was so critical for our partnership with the foundation was the long view. We've been working with the foundation, still work with them, now mostly on the clinical end, but for 20 years. So, when I remember talking to the then head of the foundation, Bob Bell, and his vision was, we're going to fix CF, and as long as you're making scientific progress, we will continue to support you. And that opens up thinking and creativity in ways that I'm still astounded by this today. We're still resonating with that type of language. The other thing that's not well known about the collaboration is that the initial big bolus of money to start it actually came from the Gates Foundation. And it was one of the rare examples of the Gates Foundation giving to a non-global health problem. And that was because there was an individual, an executive at Microsoft who had 2 daughters with CF. And I had a great privilege and pleasure of actually meeting him a few weeks ago, and his daughters are both benefiting from the therapy. So, these cycles take a long time, from the giving to the getting and then to the giving back, but they're certainly worth it, and I think a chance to reflect today, how do we make that happen more often and more quickly.
Thank you, Paul. In a sense, all 5 of you, because I'm going to think for a moment about Vertex and CF as a partnership that's gone along, have both the opportunity and the constraint of having a particular focus, particular disease. So how do you feel your organizations do in evaluating opportunities and seeking the largest impact, because in principle you have the freedom that we've said the shorter term, quarter to quarter profit-making institutions may have different constraints. So I'd be interested in, and perhaps we'll get some heat by differing opinions, and we don't all have to agree, but I'd be interested to hear specific thoughts that you guys might have about how effective is your evaluation of what makes an impact, and are you feel like you're living up to that potential? Maria, we can start with you, and then
Thank you very much.
it's
So-
free for all.
What we actually we have metrics that we actually look at.
I was
And-
going to ask about metrics, but
And so
we have to
I'll
believe in
start it off by
numbers.
saying the framework was the strategic plan. And the strategic plan was, one, bring into the pipeline strong science that has a possibility of addressing unmet medical need. So we set a number. 50 is a good number. In 5 years, 50 new candidates. And you'll see a theme here. If you can bring those discoveries all the way through successfully, a proportion of them should lead to high-quality clinical trials. So we had a target of 50 clinical trials in 5 years. Another part of the theme, the development timeline, it was something that we tested the assumption that it should take 10 years to develop a therapeutic, especially for the types of disease indications that we're looking at with cell and gene therapy. Where often they're rare diseases, small targets, maybe smaller trials.
Do you feel CIRM, on the one hand, was not restricted to a particular therapeutic area, and in a sense, every pathology was available to you, but at the same time, you have forged new criteria for what does it mean to define a
That's
novel
right.
class of therapeutics?
I think that's a very good question. So we're disease area agnostic, science-driven, and the parameters really just surround that it would be the types of programs that others would not otherwise fund. In that case, in the early days especially, it was with stem cell-based programs, highly risky, as well as now gene therapy programs, which are now there's proof of concept for that with
They're
CAR
less
Ts.
risky?
But in those days, even CAR Ts were still extremely risky. And so with that as a parameter, and then the third piece of it is that it addresses an unmet medical need for which there are no other. So we are not going to compete with a small molecule that does this better. In fact, we will fund small molecules that will recruit the regenerative potential through action on stem cells. And that's, the osteoarthritis trials in terms of inducing regeneration through the transcription factor that induces the chondroprogenitors to proliferate and make cartilage. So in terms of the metrics, what we've had in the past 3 years is now we've added 33 new clinical trials of the 50. We brought over 30 new candidates into the pipeline. We've increased the progression of programs going from basic to translational to clinical by 110%. And we've had, and our another 50 is cut development time in half, cut it by 50%. And so in order to do that, we chunked it up in terms of getting the IND enabling work done in within 18 months to IND. And so just in the past year alone, we had 4 programs go from pre-IND, where the investigators got input from the FDA, to a full-blown IND. They're now ready to start a clinical trial. They come in for our clinical awards. And then, and the way we allocate funding is milestone-based. And so we can actually track how they meet their milestones. And some things don't work, and in those cases, the money can come back so we can fund other programs. So those are the metrics that we look at.
Thank you. Yes, sir.
I'd love to maybe continue to expand on the metrics thing, but I did want to maybe talk a little bit about the other question you
Good,
asked
Brian.
about sort of impact investment and how, as a nonprofit, you sort of think about that, and was some of the experience that we've had. So when we first started in sort of the early 2000s, a lot of the work we did was, I would sort of consider more traditional grant giving. We'd launch a few RFAs every year. We would, get a bunch of proposals, we review them, give the money out to the best ideas that we could find. And you can do that, and that's sustainable. And
it's
but
not
you're
changing the landscape.
Yeah, but are you really changing the landscape? Certainly, you're giving some ideas a chance to have a little bit of an opportunity to move forward and get some answers to those. But so we really had to think a lot about the other kinds of impacts we could have. And I would have two examples I think where we've had. One is a sort of more of a financial one, but another one is I think more of a sort of infrastructural one. Mostly through the start of a few projects that we had funded where we realized the investigators, it was sort of part of a consortium effort, realized they needed some basic tools, some research tools, some antibodies to do some specific studies on particular interesting sort of targets of interest. We started actually making the tools for them. So rather than giving them the money and having them sort of go through their labs and do it, we said, you know what, we're just going to go to ABCAM or whatever.
You were
CRO.
building platforms.
We were actually making the tools for them, and these were not particularly expensive efforts. You can make an antibody for a few tens of thousands of dollars. It wasn't a huge investment from our side, but that one tool we could give to hundreds and hundreds and hundreds of labs and help them accelerate their research. So we found that one fairly straightforward way of having an impact on a larger group of the community that we were trying to support and serve was just by helping them to make some of those tools and again, make them available, either at reduced cost in some cases, certainly for some of our for-profit partners, making sure that they didn't have to pay exorbitant license fees just to get access to an antibody, things like that, our research model. So we found that actually ended up being a big part of some of our portfolio and how we could actually help sort of support a broader need that the community had. Another sort of less sort of financial tool one was more in the clinical space where we realized a lot of clinical trials in Parkinson's, and this isn't specific to Parkinson's, but certainly it was an issue for us, were failing or because they couldn't recruit in clinical trials. They couldn't get enough people. Right. So we realized that could be another way that the foundation could get involved in helping, again, a broader challenge.
And the CF has been very effective.
Exactly, about engaging people, getting them educated. Right. We created our own sort of online tool called Fox Trial Finder to help connect people, sort of, Match.com for trials for Parkinson's. So all these opportunities, strategic guidance we could give to companies, especially companies that are just getting into the Parkinson's space. We now have a whole sort of dedicated team that are sort of focused on how do you what are the unique aspects of recruiting people with Parkinson's disease into clinical trials, and we can sort of talk to companies and give them sort of more guidance. And again, I think these are, not traditional grants that you're giving to a research lab or a company to do specific work, but I think have equal sort of impact on the overall challenge. And so I think you have to sort of think creatively when you think about that type of impact investment.
Trevor, if you would, and then Ramy.
Chris.
And then I suggest we get questions from the floor, even though, so that I'm not just the only one trying to get something out of this fantastic panel. Trevor.
So Chris, just in terms of bringing some, I think, good business practices to global health, one of our biggest investments has been in the areas of measurement, metrics. And that's been mostly via this Institute for Health Metrics and Evaluation Chris Murray runs for us up in Seattle, that publishes the global burden of disease every year, which gives us a sense of what are the important entities out there. And it came from how we look at our portfolio. We took this expected net present value estimate that people do in the private sector around portfolios, and we replaced that by what we call dollars per DALY. And that's per DALY averted, that is. So a DALY is a disability adjusted life year. So that's loss of life or disability from some affliction. And so how many dollars does it cost to avert one DALY? And as some metrics that we have, for instance, male surgical circumcision to prevent transmission of HIV is actually cost saving. The bed nets that I was talking about for malaria, that's about $100 per DALY, and that's very cost effective. So $100 per DALY if something's in that range, and it's not precise, but a few $100s per DALY is an effective range. But we've seen some things, proposals. We had a hookworm vaccine that was sitting at $10,000 per DALY when I looked at the portfolio. And the reason for that is it's compared to what options there are out there. It was being compared to the very cheap drug albendazole, which does a great job of deworming kids when they get it once a year. So if you compare a vaccine which didn't exist and which was, problematic in terms of having 60% efficacy against a worm, it didn't look that good. So we got rid of that from the portfolio. So I think that you do need to measure these things because people want to understand why you're working on this and not that. And I think that there are reasonable ways that you can, even in one portfolio that encompasses devices, drugs, vaccines, a host of things, vector control products, you can still compare them in some rational way.
In fact, 4 of the 5 of you on this panel have had some long industry experience, so you understand the tension of creativity and evaluation. Ramy, just a quick comment, and let's see if there are questions from the floor.
Yeah, no, I just wanted to add that part of what we're focusing on is high risks. Again, with pharma and industry in general, you will not prioritize high risky or risky projects as your number one focus. But for us, that's exactly what we want to do. We don't believe that there's anything that's negative. If something doesn't work, that's actual data that informs the field. So when we start thinking about clinical trials, we prioritize pancreatic cancer, which is a very aggressive disease. It will not be the number one choice. We're thinking about cellular therapy. We're doing a study in kids with midline gliomas, which is right now there's no therapy. But at the same time, there's only 300 patients. So it doesn't have a commercial value. So for us, we feel that those would be the areas where we should put our resources to try to come up with the therapies for.
Great. From the floor, could we get a microphone to right over here? Please.
Thanks a lot. Good morning.
Could you tell us who's asking the question?
Yeah, sure. So I'm Robert Pacifici. I'm the Chief Scientific Officer for CHDI, which is similar to Michael J. Fox, working on accelerating therapies for Huntington's disease. And I got to say, I'm really impressed with what I've heard all throughout the morning, including the panel. But I'm a bit surprised at something that I haven't heard yet. So I think, if you take caliber as an example, I'm sure you don't want to take that really well-oiled machine and produce potent, selective, well-tolerated compounds that are not efficacious. And so I kind of wanted to riff off of what Pete Schultz mentioned this morning, which I think is true. It's the golden age of biology. So if you operate off the thesis that the single most precious thing to a drug hunter is an observation made in the population you seek to treat, I think that one of the things certainly that we've tried to focus on at CHDI is experimental medicine, to really learn more at a more granular level what's going wrong with these diseases so that we can figure out which targets to prosecute, what things to measure, what mechanisms are in play. And I wonder whether folks agree and what efforts are being put into experimental medicine to reverse translate.
If I could just offer a comment, and then I'm going to speak for one of your colleagues, David Altshuler, who's at Vertex, and who has a giant distrust of animal models and believes only if there's some genetic and pathologic indication in human subjects that it's worth the investment of time and effort. I'm sure I'm paraphrasing him incorrectly, but that is how I think he used to think. Sir.
So 100% agree. I mean, the best animal model of disease is the human with the disease. So we've taken that to heart as well and put a lot of effort and support around ways we can best study Parkinson's disease in people with Parkinson's. Probably our biggest investment is we have a longitudinal cohort study that we've been funding for about 10 years now called Parkinson's Progression Marker Initiative. These people who are just amazing participants because they let us poke and prod them in every which way that we can, and neuroimaging and blood samples and urine samples and spinal taps. So they get, at the start of the study, they're getting multiple spinal taps in the same year, and then every year getting another tap. So the fact that they even will undergo that, I think, is amazing. Right. Really creating a good resource for trying to better understand at the biochemical level, but also clinical level and digital. We're now wearing watches on all the individuals. So all the different ways that you can sort of measure the disease, really to answer that one specific question, which is what goes wrong in people with Parkinson's disease that might lead to their disease so that we know what we need to target to fix. And so I think that's really, really, really critical.
And of course, Pete mentioned in his talk this morning that Eric Topol will give the third major talk of the conference today, I think at 5:30, and that is very much an epidemiologic population-based approach of genomics and genetics around humans.
guess I would just add to Bob's comment that I think CHDI is one of the organizations who's leading the way in trying to do these longitudinal assessments of human patients and how they progress to the disease and what are some of the earliest markers that the disease is beginning to occur. Because we know for some of the neurodegenerative especially that, but maybe for most genetic diseases in fact, that the earlier you treat the better. So you want to be able to get into patients before they're too far advanced with some indication that's relatively simple and quick to do in the clinic that you're getting efficacy. So I think that's one of the things that the not-for-profits can really do, and it's good to hear the Michael J. Fox Foundation and of course the CF Foundation was also a pioneer in that. And having patient registries as well, so that the patients are signed up, you know where they are, when the therapy comes along, you can bring it in quickly to the population.
Question, second question, or
Yeah.
another question.
I have a question about academic research and the problem of reproducibility. We all know that that's an issue and that VCs and companies certainly have the resources to try to reproduce the experiments before they in-license things.
You can never publish in Nature or Science.
Yeah. So how do you guys deal with that?
Well, happy to start. I mean, this is, I think, a really important challenge to try to address. I mean, we approach it in a lot of different ways. You know, sometimes, depending on the question maybe we're trying to look at, we might build it from the start as kind of a consortium type of approach where we have multiple groups sort of testing the same idea, you know, the hope that they'll all come up with the same answer. In other cases where it's something gets published in Science or Nature, and the idea is suddenly everybody thinks it's the newest thing, and how do you kind of get enough clarity around it? So we will occasionally then do kind of replication studies where we'll support someone else to try to repeat that work. I think it's challenging because it's we get sort of caught up in, I think, the terminology here. You know, there's sort of replication in the sense that you're going to do the exact same experiment that the person did, following exactly the same protocols and everything else, versus is it really just reproducing the finding, which might just be something more straightforward, which is, you know, with a variety of other techniques, can you actually observe the same change in biology, whatever it is you're trying to reproduce. So I think it's just making sure that you're sort of clear about what it is you're actually trying to reproduce and replicate, and then developing your kind of funding approach around that.
Trevor?
Karen, there's a variant of that problem in the global health space, which is that there's certain interventions which folks working in global health love, and for good reasons. For instance, breastfeeding. Exclusive breastfeeding for 6 months is said to be the panacea. It cures almost all ills, the stunting that I talked about. Well, it's not exactly true, actually. And if you look at the varying studies of breastfeeding, it depends about what you're talking about. So there's the notion that breastfeeding reduces diarrheal disease in kids. True, strong evidence for that. There's the notion that breastfeeding reduces upper respiratory tract infections. Turns out very weak evidence. So we've been funding an effort now to go back to all of these nostrums about this is great, you know, chlorhexidine for umbilical cord care is universally good. To take some of these interventions that people just believe in and look at the spectrum of evidence, and with a system of weighing the unexplained variability. So that's how you'd put it.
Yes.
So what is the unexplained variability in the results? And that gets you a negative rating. If you can explain it by covariates that you understand, that's fine. But if they're unexplained, that gets you a negative rating. So it's almost like a consumer report. You got 5 stars, you got 1 star evidence. 5 stars, cancer, smoking and lung cancer. 1 star, even less, omega-3s and any health outcome, just to give you a preview of before that work comes out.
I just maybe one comment. I do think that there's a broader challenge in the sort of whole replication concept too, that, you know, given all the investment we go to get to the first discoveries in the first place, to then repeat those investments to duplicate everything to see what stuff is true and what isn't, I think it's a real challenge. So I think there's an area and a need for kind of more innovation on different ways to quote unquote replicate the findings. And so whether it's again the use of creative data mining and sort of big data science type of approaches, the data may exist out there and you just have to sort of find it in other ways. But, you know, I think also thinking creatively about how you go about doing that replication, I think is important, because there's just no way we could.
And maybe everyone in the community needs to be a better reviewer of those claims and papers. Other questions or comments? You can make a quick comment. Yes, sir. Pete Schultz, you have anything to say? No? That's unlike you.
So just the reproducibility thing, one thing that we've tried to do is to be proactive about it. So rather than get a result and say, I wonder if I believe it, we actually now participate in the design of the experiment and the statistical plan up front. This way we can be sure that it's properly powered, the right kinds of analysis, Bonferroni corrections, all that good stuff. The reality of it is most of us, I know in my training, I wasn't properly trained as a statistician, so people guess on the design of studies and then try and extract too much of them post-hoc. So.
One of the good things about the Comprehensive Cancer Center Network is that they all have biostatisticians who are part of the community that evaluate any potential trial going forward. Let me ask a question of the panel, especially those of you who've had experience in industry. And you've certainly internalized many of those insights into the current perspectives you take in these not-for-profit or, in Vertex's case, reasonably profitable. How does your perspective differ from when you were running programs? That is, in principle, I could argue that nonprofit foundations and institutions of the type you represent sit at a different niche along the spectrum of how does one facilitate biomedical research, especially in a translational mode. Are there pieces of wisdom that one could, pithy, that one could enunciate that reflect different points of view that you now have that were different from when you were in industry? Trevor, let's start with you.
Well, Chris, I'll go to a problem that I had when I joined Gates, and I tried to recruit some folks out of industry to join me there. And their initial response almost uniformly was, well, you folks are just bankers. So, we're active people. We actually do things, and you're just bankers. So
That's a
we
certain
wouldn't be
conceit.
interested
Yes.
in your kind of activity. And I think, that's the...
But I'm making the assumption that you are all activists.
Yeah.
Activist investors rather than passive bankers.
I think, and that's a message that I would say is that that's been the sea change that I've seen in our institution, is that we don't have a divide. We do whatever it takes, however activist we have to be for programs to work, doing basic project management, which is always essential. To, in some cases, where we've got a great partner, often a private sector partner, that can do things themselves, and then they can run with the funding. But if we don't, then we have to make the pieces fit, and that's been the big change, I think.
So I think Brian and Paul have both said, in some sense, platform building and taking the long view seems different from the kind of yearly review of programs that I know in large pharma. Paul, any?
Well, I've only lived at Vertex, so I don't know what goes on at other pharmas, but I think there is a change. At least the experience with CF was transformational for us in the sense that it provided evidence that you have to take the long view to be successful, and your job then as a company is to survive long enough to execute on that long-term plan.
Good.
And I think when you look at it that way, and I think more companies are, at least the people I've talked to are, I think there's greater opportunity now to speak a common language. And while yes, the yearly cycles occur, I'm sure they occur at all of your organizations. It's just making sure you're keeping your eye on the long view, and that you have milestones that can measure your success toward that goal. And those can be yearly, they can be monthly even. And so it's a way of measuring progress. And I think sometimes, just number of new chemical entities into the clinic or into phase 1, 2, or 3, those maybe are not the metrics. They are convenient, but they're not deep.
I'm going to give Pete the last word, and we're going to stay on time. But I do want to say that I think each of you in your own ways, not only as excellent representatives of institutions, are very much educators in terms of your clients are really the academics who are interested in moving beyond the Science or Nature paper and the IP, but really learning the process of what translation and drug delivery means. And so I think you should feel good about your ambassadorial roles. Pete, last comment.
Yeah, no, is my experience in kind of marrying basic and translational research is there's this view that you really can't work in a field effectively unless you have a lot of experience in that field, and you have to be one of the grand old men or women in that field. But what you see...
You're getting older all the time.
And I'm excluded from that, okay? So what you see is, at least when we built GNF in Caliber, is we hired a lot of really amazing young people that were just looking to do something interesting and exciting. And it was amazing how fast not only did they learn preclinical drug discovery, but how fast they're learning clinical drug discovery. And I think one of the problems in the academic research field is people expect you to know how to do something before you do it. Whereas I think in this area, if you can begin to capture the young folks and the energy and intellect that the young folks have, and how do you think about that? Because when you review grants, is it like, this person's going to do glioblastoma, but they don't know anything about glioblastoma, so why would we fund them? And how do you think about engaging the young generation with all of these challenges? I'm just curious, because I think it's something the NIH doesn't do so well.
So, of course, it's such a great question, we're out of time. I do want to thank the panel and the audience for this hour, and I hope that my colleagues on the panel will be around through the afternoon and the 5 to 5:30 time, so you can buttonhole them for questions you didn't get to ask in this hour. Thanks very much.